Thank you, everyone from around the world. My name is Lorraine Resch, Vice President of Scientific Strategy and Commercial Development. Some housekeeping items. Our estimated time is 40 minutes plus Q&A. Remember to submit the questions via the Q&A button, and we'll address as many questions as time permits. Any questions that we do not have a chance to answer, someone from Celerion will address directly within 24 to 48 hours. Today, we will discuss the quality of sampling, preparation, and analysis and how it can affect flow cytometry data. We will evaluate how on-site clinical sample collection and on-site bioanalysis laboratory processing in real time enhances the quality of data. Today, we are fortunate to have Anamica Muruganandham sharing her scientific expertise to help position you, our clients, towards a successful understanding of how improving PBMC flow cytometry results supports exploratory and early phase clinical trial endpoints. Anamica is a bioanalytical senior scientist in molecular and cell biology at Celerion, where she focuses on method development and validation of qPCR, flow cytometry, and immunogenicity assays. She has led the recent development and validation of several COVID-19 qPCR and antibody assays used in Celerion clinics to assess subject and employee safety in clinical trials to support drug and vaccine development endpoints. She is also currently leading the expansion of COVID-19 PCR testing to the Phoenix Celerion laboratory. Additionally, Anamica is a subject matter expert leading the team for PBMC isolation techniques, serving as a bioanalytical project leader performing data analysis for qPCR and flow cytometry studies. Anamica joined Celerion in 2016 after obtaining her master's degree from the University of Houston, Texas, and is gaining increasing tenure in the bioanalytical team at Celerion. And with that, I turn it over to Anamica.
Thank you, Lorraine, for the introduction, and thank you to everyone for joining today. Celerion has several years of experience with PBMC isolation and flow cytometry studies to provide clients with important data pertaining to immune response. Today, I will provide insight into our PBMC isolation and flow cytometry capabilities that is important in early drug development. We also have a bioanalytical lab in Zurich, Switzerland, where we are expanding and adding flow cytometry capabilities, which will be open in Q1 of 2021. We are also implementing PBMC capabilities in our Phoenix, Arizona site. So today, first, we will look into PBMC isolation, which is peripheral blood mononuclear cells. So what are PBMCs? Peripheral blood mononuclear cell, which is called as PBMC, is any blood cell with a round nuclei, such as lymphocytes, which consists of our T, B, and natural killer cells, monocytes, and dendritic cells. They are critical components of the immune system. They elicit an immune response using several signaling molecular pathways. Isolation of PBMC is commonly used to measure response to certain stimuli via a variety of technologies, including cytometry, ELISpot, which is enzyme-linked immune absorbance spot, and qPCR, quantitative or real-time polymerase chain reaction, which is widely used in the current situation of COVID. So how is PBMC isolation done? Isolating PBMC in a quality manner is very critical to investigating the responsiveness of the immune system to a treatment. There are three types of techniques using density gradient separation that can be used for PBMC isolation. They are underlay technique, overlay technique, and separation tube technique. We will go through each technique in the upcoming slides. Celerion analysts have several years of experience and expertise to optimize PBMC isolation methods to meet the demands of the downstream application from the clients. All PBMC methods are custom qualified for the end application under the supervision of an experienced scientist. We do have an internal method where we qualify all our analysts first before they are allowed to qualify on a client-based method. Clients sometimes have requirements that they need to see our capabilities by analyzing our qualification samples to ensure that they have enough cell count and viability for their downstream application. Therefore, we ship qualification samples to check for viability and recovery to the clients for their downstream application as part of site qualification and analyst qualification. So the next technique that we're going to see for PBMC isolation is separation tube technique. The image to the right shows the blood aliquoted and a phycol layer right below the blood, which is separated by a frit. The frit is a porous barrier or a gel that provides extra separation between the red blood cells and PBMC layers. We have experience using several different kinds of separation tubes, which are leukosept tubes, sepmate tubes, and cell preparation tubes. Diluted or undiluted blood is slowly aliquoted by pipetting it down the side of the tube, where the blood will be layered right on top of the frit. PBMC isolation can be done with blood that is collected from several different anticoagulants, such as EDTA, sodium heparin, lithium heparin, and sodium citrate that is collected fresh or within 24 hours. For sample shipping, it is recommended to use CPT tubes, which is the cell preparation tubes. They can be centrifuged at the clinic site and shipped on cold packs overnight to perform PBMC isolation and cryopreservation. CPT tubes are expensive, but they are designed for overnight shipping, which allows the benefit of shipping samples to labs, especially with a multi-site study to ensure similar quality of PBMC isolation and recovery. So in this slide, we are going to see how PBMC sample is handled once we receive it at Celerion. Since PBMC isolation is time sensitive, we have a process outlined for handling of samples from sample receipt to cryopreservation. We require at least 1 week of notice for upcoming samples prior to sample arrival. This ensures that we can schedule appropriate resources for smooth processing. Sample then arrives to the lab or sample management within 30 minutes to 24 hours, depending on the location of the clinic site. Since Celerion has a clinic on site, we are able to receive samples within 30 minutes to our lab, and it can be processed right away. Samples are then immediately checked for paperwork and signed for receipt. Samples are processed immediately, documented in lab notes, which is our electronic notebook system, following the appropriate protocol. In our electronic notebook system, we record the reagents used, sample when the time was received, when the sample was started processing, and when the processing ended. By this way, we can know that how long it took for processing the samples, as well as what kind of reagents was used for processing. The samples are then placed in a cool cell or minus 80 degrees. Cool cell is used for slow freezing of cells at a controlled rate of -1 degree Celsius per minute to prevent ice crystals from forming, as they can affect cell viability. Samples are then transferred to liquid nitrogen if needed. Samples are shipped in cryoport or dry ice based on client requirements. If we follow our internal method, we have the appropriate bioanalytical method procedure already recorded in lab notes. If there is a client-specific method, we make a client-specific bioanalytical method procedure using the manual provided, and it will be sent to the client for review before uploading to lab notes. So in this slide, we will see PBMC sample processing using Celerion method. So first, we can collect 15 to 30 ml of blood using EDTA or heparin as anticoagulant. The amount of blood can be adjusted based on the requirement of the assay. Then the blood is transferred undiluted or diluted to leukosept centrifuge tube. The blood is then fractionated by density gradient centrifugation with phycol PEG+. The image to the right shows how the layers are formed after density gradient centrifugation. First layer is plasma. Then you can see a white cloudy layer, which is our PBMCs, followed by phycol medium. Then we have the porous barrier. Below it, we have our erythrocytes, red blood cells, and granulocytes. After centrifugation, the plasma is collected and frozen at -80 if the assay requires collection of plasma. Most of the clients suggest that we collect plasma because they are similar to plasma that is isolated during centrifugation after blood collection. By this way, we can obtain both PBMC and plasma collection from a single sample. That can reduce the amount of blood that is collected from a participant. Then we harvest the PBMCs, wash and count the PBMC, and cryopreserve the PBMC in liquid nitrogen. So we are going to see how we count PBMCs. We use an instrument called Muse Cell Analyzer. It is a compact instrument for fluorescence-based 3-parameter cell analysis. The 3 parameters being live, dead, and cell size. The Muse count and viability reagent stains viable and non-viable cells based on their permeability to the 2 DNA binding dyes, which are a DNA binding dye and a membrane permeant DNA staining dye. A deoxyribonucleic acid DNA binding dye stains cells that have lost their membrane integrity, which allows the dye to stain the nucleus of dead and dying cells. A membrane permeant DNA staining dye stains all cells with a nucleus. This helps to discriminate cells with a nucleus from debris and non-nucleated cells, such as your red blood cells. Here is an example of PBMC counting report, which is displayed to the right, and that report is generated from our Muse Cell Analyzer. There are 2 profiles that is generated from this instrument. To the left, we can see the population profile, which is based on cell size and a viability profile. The threshold line, which is the dotted line of the cell size index, is mainly set right below the first population, as that contains your PBMC, and the cells below the line represent platelets, debris, and non-nucleated cells. The viability profile shows live versus dead cell percentages of nucleated cells. Any cells that is seen closer to the threshold line are apoptotic or dying cells. The report also provides viable cells per ml, percent viability, total cells per ml, total viable cells in original sample, and total cells in original sample along with dilution factor, and it is calculated based on the original volume which has been used for PBMC cell counting. Celerion analysts routinely achieve 1 to 2 million cells per ml of whole blood sample from a healthy donor. Deceased blood counts can vary from 0.5 to 2 million cells per ml of blood collected. Viability of isolated PBMCs is a very key parameter. Our analysts routinely achieve 95% of viability of isolated PBMCs. Here we show the stability of PBMC post-thawing. Cell count and viability after thawing is very important for performing downstream applications such as flow cytometry, qPCR, and ELISpot. It is important as you need live cells and a certain amount of cells to perform the downstream assays. Celerion method shows high recovery and high viability rates post-thawing. The graph to the right represents percent viability versus days stored in liquid nitrogen, and you can see that the percent viability up to 252 days maintains at 90% viability. And the image below it, the cytometric analysis dot plot of PBMCs, which is analyzed 237 days post-collection, shows very distinct population of debris, which is to your left, around 100K profile, you can see lymphocytes, and the blue population to your right represents your monocytes. So this shows that our viability and recovery is very high, and we can get high purity of PBMC, and they can be stable for up to 252 days. Plus, our recovery rate is achieved at 70%. So for example, if 10 million cells were frozen or cryopreserved, we are able to obtain 7 million cells, which is 70% recovery. So next, we are going to go into flow cytometry capabilities. PBMC is one of the common sample types that are used for flow cytometry processing. What is flow cytometry? Flow cytometry is a technology that simultaneously measures and analyzes multiple physical characteristics of cells as they flow in a fluid stream through a beam of light. The image to the right shows a cell suspension, which is labeled with a fluorochrome, that enters the flow chamber, and then as it exits, the cells pass through a single file of droplets that are formed due to hydrodynamic focusing by the sheath fluid that surrounds and narrows the sample to flow through a single file. This reduces protein buildup, cell accumulation, and keeps cells in the center as it passes through the laser beam. As it passes through the laser beam, the light is scattered based on the cell type, absorbed, and emitted at different wavelengths, which is captured by specific detectors in the instrument, and the data is sent down to the software. An application of flow cytometry is cell sorting, which we will see in later slides, but briefly, it is the process that you can see in the image where certain cell types of interest are collected and sorted into separate collection tubes for further analysis. Flow cytometry panels can be customized based on the markers required to distinguish specific and rare subsets. The properties that are typically measured with flow cytometry is size, granularity or complexity, and fluorescence intensity. What are the advantages of flow cytometry? It can measure single and large number of cells. It can simultaneously analyze multiple parameters. Sorting of defined cell populations can be performed. This technology helps with understanding the immune response to a drug or vaccine. We can quantitate by fluorescence intensity signals. It can also identify small and rare populations. So in the next few slides, we are going to look into the instruments used at Celerion for flow sample acquisition and their features. We have two FACSCaliber DX P6 instrument, which is a FACSCaliber that has been refurbished by Cytec to modify the optical configuration to include two lasers, red and blue, with 6-color analysis that provides flexibility to support a wide variety of research and clinical applications. This instrument can be used for immunophenotyping, which is the analysis of heterogeneous populations of cells for the purpose of identifying the presence and proportions of various populations of interest. It can also be used for bead array assays, which is a powerful flexible tool that allows simultaneous measurements of chemokines, cytokines, immunoglobulins, or cell signaling targets from a single sample. We also have a BD FACSMelody cell sorter, which can perform immunophenotyping assays with the advantages of cell sorting. This instrument has two lasers, red and blue, with 6-color analysis for sorting cells. The sorted cells are based on high purity and recovery for downstream application, and they can be sorted into a single tube or into a 96-well plate. Cell sorting can be done based on intracellular characteristic. Cells can also be sorted by labeling with fluorescent probes for nuclear or other intracellular targets. To obtain information on cell surface molecules such as membrane protein receptors, even if they are low in density, cell sorting can also be performed according to the presence, absence, and density of the receptors. The third instrument that Celerion has is the BD-ELISA photassa, which contains 4 lasers, red, blue, violet, and ultraviolet, with 18 color analysis capability. The sample acquisition can be performed through a single tube or using a high-throughput sampler to improve experimental workflow. This instrument provides rapid, fully automated sample acquisition from 96 well plates. The sample carryover when acquired via plates or tubes is less than 0.5%, and this ensures sample purity and data integrity. Samples can also be multiplexed with various combinations of antibody staining, and they can be plated in a single plate that can provide a variety of data. In this slide, we're going to look into the parameters that are considered for a flow cytometry method development. First, we need to figure out what the purpose of the assay is by finding out the cell type and the markers of interest, whether it has been used for clinical or preclinical safety assessment or pharmacodynamics assessment. The assay format has to be designed based on the purpose of the assay. Is it going to be cell surface or intracellular staining, functional assessment or phosphoflow assay? Is it for receptor occupancy or target engagement? The type of matrix plays a huge role in designing the appropriate protocol. The volume of matrix is important to consider when designing the assay. Different types of matrix that can be used is whole blood, bone marrow, PBMC, white blood cells, tissues, cell lines, body fluids such as plasma, sputum, and cerebral spinal fluid. For certain populations such as cerebral spinal fluid and diseased population or bone marrow, it is really hard to obtain a large amount of volume. Therefore, when designing the assay, the volume that can be obtained needs to be given extra consideration when picking out the matrix. Next, how we can collect the sample. Is there a choice of anticoagulant that is preferred? Are there any additives that has to be used? Is a stabilizer or preservative agents? Can we use ready-to-use blood stabilization tubes like BCT tubes or SMART tubes? Or do we require isolation of PBMC before proceeding to flow cytometry? And we need to think about if tissues processing to cell suspension. The last thing to consider is panel design. So the panel design needs to consider the marker biology, whether it has shedding or internalization issues. Shedding or internalization of markers can cause a signal to reduce after a period of time. Antibody clones needs to be selected. Fluoropore selection needs to be done based on the requirement of brightness for the markers, low, medium, or high brightness. For example, if a population is present in high amounts, then a low brightness or less intensity fluoropore can be used. For rare population, a high brightness or high intensity fluoropore needs to be selected to detect them without any issues. The last one to consider is whether there are quality controls available commercially or does it need to be custom made. Next, we are going to see what is achievable in flow cytometry validation. There are currently no regulatory guidelines on flow cytometry validation process, although there are various publications on fit-for-purpose approaches and validation recommendations. However, whenever flow cytometry data are used in studies conducted in a regulated environment, establishing a proper validation testing protocol based on the requirements of the assay and the intended use of biomarker data is very important. So here we can see the parameters that can always, sometimes, and some of the parameters that cannot be achieved via flow cytometry. So we can achieve specificity, Antibody specificity must be determined to ensure that there is no cross-reactivity or binding to Fc receptors to avoid low-intensity signals. Precision can be performed by testing same samples by different analysts on different days if there is stability and on different instruments. Sensitivity, such as limit of detection and limit of quantitation, can be determined based on the population. For rare events, the limit of detection can be calculated as the mean of all data points plus 3 times standard deviation. Stability needs to be established, especially if samples cannot be processed right away, which we will see more in more detail in the upcoming slides. Reference intervals can be established by previously known data. In the first stages where a marker is exploratory, reference intervals are not necessary. But once data has been established, it is always better to have reference intervals in an assay. Sometimes we can achieve linearity, standard calibrators, and interference. We can figure out interference from matrix or interference from drug. Accuracy is very hard to be obtained in a flow cytometry assay due to the lack of proficiency testing programs, lack of cellular reference material. QC material might not be available for accuracy, and for novel or proprietary methods, sample exchanging is not possible. Selectivity is typically checked during method development to ensure that all the reagents and antibodies can detect the markers of interest. It is also hard to determine range of quantification. Incurred sample reanalysis cannot be performed if there is no freeze-thaw stability established. Normal signal distribution and prozone effect can also not be established for a flow cytometry assay as it is done in an LCMS assay. But we can still modify all the assays for fit for purpose to achieve the required data. We're going to see more about stability challenges in the next few slides. So stability of samples for flow cytometry is a very critical parameter, as it is restricted that the sample can only be stored to a limited period of time after collection. Sometimes clinical samples have to be shipped to a testing laboratory. If the samples are stabilized, this can allow for batch analysis of all time points from the same subjects together to reduce variability. Multiple options for stabilization include adding fixation or preservation agents upon sample collection, freezing isolated cells for extended periods of time, determining the optimal storage conditions, whether it has to be stored at room temperature or does it have to be refrigerated. Celerion has a proprietary stabilizer that can increase sample stability, allowing for the shipment of samples to Lincoln from multi-site late-stage studies. In this slide, we are going to look into stability assessment steps. Assessing stability can be done in 3 different stages from sample handling to acquisition. First stage is sample collection and shipping stability, where we evaluate the sample collection process by considering the matrix anticoagulant extra. Then we consider, is the sample stable and can it cover the required storage period prior to analysis in the context of the study? If it can, then we look into, can the samples be stored for long-term storage? If the sample is not stable, then we'd have to try different collection tubes or stabilizing agents by comparing to reference collection tubes, example EDTA or sodium heparin. Then we check if the sample needs to be collected in a fixative or a preservative for short-term or long-term storage. If that is achievable, then we need to again test for long-term storage. If in both cases long-term storage is not achieved, then the sample has to be processed immediately and can be checked for benchtop stability. If the sample cannot be collected in a preservative, then we need to test for different storage and shipping temperature conditions, such as ambient temperature 5 degrees or minus 80, and storage periods after collection prior to processing. The second stage is to test for frozen storage stability, where we can evaluate freeze-thaw stability, and we can assay samples at baselines and various time points to establish long-term stability. The third stage is processing stability, where we can evaluate for benchtop stability and stability of samples post-thawing. We can test different acquisition times after sample processing and consider fixed versus stained samples for processing. This slide shows several examples of sample stabilities that we have established at Celerion. So the first assay is the leukocytes or hematopoietic stem cell assay, which is a CD34 panel, where we have established long-term stability for 182 days at minus 80. We also have established pre-process shipping stability and post-process stability. The pre-process stability is very important, where a sample has to be shipped from a clinic site to a lab that is elsewhere. This can allow for shipping and processing of sample without losing the integrity of the markers. Post-process stability is also very important, as we need to know how fast the samples need to be acquired after staining. Some assays that have been listed in this table at the bottom, which is axial phosphorylation and mer phosphorylation, typically takes around 8 to 10 hours for staining. In those assays, it is difficult to acquire samples after staining due to long working hours and less availability of resources, especially if the lab is not 24/7. Therefore, splitting the staining and acquisition processes to 2 different days allows for easy acquisition data. So in this table, you can see that we have established post-process stability, which is samples can be acquired within 72 hours after processing and 75 hours after processing for mer phosphorylation, which gives us 3 days of time after staining to get the data. We have also established a TBNK panel where long-term stability has been established for 338 days at minus 80 and freeze-thaw stability for 4 freeze-thaw cycles. We've also established pre- and post-process stability at 22 hours and 24 hours after collection. In the upcoming slides, we will see the CD34 and TBNK stability examples and challenges that we faced when establishing those stabilities. So here we will go into detail about how the long-term stability for CD34 assay was established. So CD34 is a marker of hematopoietic stem cells. Hematopoietic stem cells are stem cells that give rise to other blood cells. They are characterized by the expression of CD34 and are relevant in a number of clinical settings, including the treatment of various leukemias. One such treatment is to administer granulocyte colony stimulating factor in order to repopulate the hematopoietic system after chemotherapy or irradiation. Current protocols using whole blood mention that the sample needs to be stained with antibody and run via flow cytometry immediately. But we have here shown a method for stabilizing the samples that can result in large number of samples to be processed in batch analysis. So for this assay, mobilized human whole blood was purchased from all cells. Celerion stabilizer was added at 20% volume of blood within 24 hours of collection, and the sample was then frozen at minus 80. The panel to the left, which is highlighted as ABCD and is highlighted by day 0, represents the fresh sample that was collected without stabilizer addition. The day 182, the panel to the right represents the analysis of the sample that was frozen for 182 days at -80. So here is the gating strategy for CD34 hematopoietic stem cells. The plot to the left under day 0, column A, represents FSC versus SSC. FSC is forward scatter versus side scatter, which separates your populations based on size versus granularity on the side scatter scale. So you can see the leukocyte profile, which contains your debris to the left, and towards the bottom of the plot, there are your lymphocytes. Towards a right upper corner, there is monocytes, and on top of it, we have the granulocytes. The beads are then gated, which is your plot B. Plot C represents all CD45 positive events, which is your leukocyte population. Plot D represents further gating of CD45 population to gate CD34 hematopoietic stem cells. As you can see, from day 0 to 182, the populations of interest, which is CD45 and CD34, are very similar, and the percentages are very similar as well. From day 0, CD34 at 0.28% versus day 182, 0.30%. When looking at the dot plots, it is also noticed that the positioning of the leukocytes shifted with a decrease in CD45 intensity. We also noticed under day 182, plot C, that there is an increase in the debris population. We did not investigate that, as we could not see it causing any problems in gating our population of interest. There was not much interference from those extra population for the stability experiment. But due to the shift in leukocyte, with the extra debris population arising from day 82 as represented in image C, the gating was adjusted for day 182 to exclude additional debris. But we were still able to gate CD45 and CD34 populations distinctly, and their percentages were mostly unaffected by the stabilization process. This slide shows a graphical representation to show the percent difference from day 0 when samples were tested on days 7, 14, 30, 49, 70, 90, and day 182. The absolute CD34 positive cells per microliter for long-term stability sample closely matched the day 0 sample value, passing the acceptance criteria of plus or minus 30%. Next, we are going to look into how freeze-thaw stability was established for TBNK markers at Celerion and the difficulties that was faced during this stability challenge. Lymphocyte immunophenotyping of T, B, and natural killer, which is your NK cells, helps in providing valuable information regarding a person's immune response. In this assay, we differentiate T cells to two kinds, helper T cells and cytotoxic T cells. Helper T cells help other cells in the immune system to start and control the immune response against foreign substances. Killer or cytotoxic T cells helps with clearing out infected or malignant cells. B cells make antibodies to fight foreign substances known as antigens. NK cells also respond to foreign substances and are specialized in killing cancer cells and virus-infected cells. Therefore, we wanted to test if the sample can be freeze-thawed to allow for repeat analysis if a sample needs to be tested again or if we need to obtain additional data. So the image to the left represents control sample, which has 8 different plots from A through H. The panel to the right represents the data after freeze-thawing the samples 4 times, and the images go from A through H. So in the control panel, if you can see in plot A, we see FSC versus HSC cells distribution for whole blood. In image B, the beads were gated out. Image C shows gating of lymphocytes, which is further differentiated into T lymphocytes of CD3 plus and CD3 minus markers. The T lymphocytes from image D is further differentiated into images E and F, which is differentiated into the 2 different kinds of T cells, which is helper T lymphocytes and cytotoxic T lymphocytes. The CD3 negative T lymphocytes from image D is gated further to represent the B lymphocytes, which can be seen in image G. And image H, CD3 T lymphocytes is again further gated to represent natural killer lymphocytes. Samples were analyzed using stabilized frozen whole blood using our Celerion stabilizer. The control panel samples did not go through any freeze-thaw cycle under white light condition. The panel to the right, which is FT4, was analyzed after samples underwent 4 freeze-thaw cycles under white light condition. Human whole blood healthy donors were collected at Celerion, and our stabilizer was added at 20% volume to the volume of blood, which was stored at -80 and underwent the 4 freeze-thaw cycles. Even in this freeze-thaw, the image to the right, freeze-thaw 4, image C, plot C, shows an increase in the debris population compared to the control plot C. This was not further investigated as the sample integrity for all lymphocyte subsets were unaffected by the freeze-thaw cycles. We are still able to gate the lymphocyte population without any interference from the increase in debris noted in freeze-thaw 4 samples. This shows the graphical representation of the TBNK freeze-thaw stability. The percent difference from control can be seen that it is within acceptance criteria specifications of plus or minus 25% of the control sample for all different subsets of T cells, TH, which is your T helper cells, TC, which is your cytotoxic T cells, B cells, NK, which is your natural killer cells. So this slide represents the sample handling, how it is performed when samples are received at Celerion. The bioanalytical principal investigator, BPI, who is in charge for the entire study, receives notification of upcoming samples 48 hours to 10 days prior to sample arrival. Samples then arrives to sample management within 30 minutes to 24 hours, depending on the location of the clinic site. Samples are immediately checked into Watson by sample management. Watson is our LIMS-based system that keeps track of samples received, analyzed, which samples are not analyzed, sample discarding status, etc. If stability has been established for the assay, sample has stabilizer added and stored in appropriate storage condition. If there is no stability, the samples are processed immediately. Once the samples are received in sample management, the work list is made in Watson by bioanalytical project leader, and samples are backed by sample management. Work lists are typically prepared based on how the samples are analyzed. All time points for a subject are analyzed together, as that gives valuable data when analyzed in a single batch, which has less variability in terms of processing, instrumentation, and sample handling. The analyst then checks out samples from sample management and verifies for order and accuracy against the work list. Samples are aliquoted and returned to sample management for long-term storage. The samples are then processed according to bioanalytical method SOP and documented in lab notes, which is our electronic notebook system. The flow data is then analyzed by bioanalytical project leader. There is a secondary review of data and gating strategy performed by another experienced project leader or an experienced scientist, and the final review of data is done by quality control team. Next, we will look into intracellular staining. Intracellular staining is very important part of flow cytometry assays. So flow cytometry is used to analyze various intracellular markers such as phosphorylated signaling proteins and cytokines. Celerion has several years of experience performing intracellular staining by fixing, permeabilizing, and staining cells with antibodies. The fixing and permeabilization treatment allows the antibody to pass through the membrane, get to the interior of the cells while maintaining the morphology of cells. Analysts have expertise in culturing cells and passaging of cells. Samples for intracellular staining are typically analyzed based on median fluorescence intensity values, and phosphorylation changes can be calculated using reference samples, which are your pre-dose samples. Median fluorescence intensity value shows the level of antibody expression. This slide shows drug-induced inhibition of phosphorylation that was performed for a study at Celerion. This assay used plasma samples collected from dosed individuals to measure the inhibition of phosphorylation to certain intracellular markers. The cells were fixed, permeabilized, and stained before acquisition. So the image to the left, phosphorylation inhibited cells, shows the dot plot FSC versus SSC scale of phosphorylation inhibited cells. The plot to the right, which is blue FL2 versus count, shows the histogram of phosphorylated versus inhibited cells. Since we are detecting phosphorylated inhibited cells, we can see a shift in signal to the left, which shows high intensity signal and high expression of antibodies. The graph below of percent inhibition versus drug concentration shows how inhibition of phosphorylation is happening with respect to drug concentration. With increase in drug concentration, there is increase in inhibition. In this study, the highest drug concentration of 10 micromolar shows 100% inhibition of phosphorylation. This kind of assay can provide really important information in terms of oncology drug development, as inhibition of phosphorylation also helps with preventing cell replication, mainly cancer cells. Cell sorting. Cell sorting is very important in clinical diagnostic and research and is an important feature of flow cytometry technology. It also allows to isolate and collect individual types of cells from a heterogeneous mixture of cells. The cells are sorted by deflecting the appropriate populations into collection tubes or to perform index sorting based on the light signals emitted. Index sorting is when a single cell is sorted into a single well of a 96 well plate to perform further analysis. Sorting can depend on cell surface or intracellular characteristics of a cell. Celerion has experience sorting cells using BD FACSMelody to collect high purity cells. Here is an example of cell sorting using our BD FACSMelody system. The first plot, SSC versus CD45, shows acute myeloid leukemia cells, myeloblasts, which is gated in the box, which has the red and black populations. The myeloblasts are further gated to identify myeloid cells, which is CD13 and CD33 positive cells. This population is further gated in the sorting plot and represented by a quadrant gating using CD117 and CD34 markers. AML, or acute myeloid leukemia specific cells, have CD34 and CD117 markers. There are 7 different classifications of AML according to World Health Organization, and the sorting needs to be performed based on CD45 SSC profile and CD34 CD117 profile, as some classifications are CD45 negative, and some classifications can be present in any one of the 4 quadrants represented in the sorting profile by being double positive, double negative, or single positives. In this example, the cells that we are sorting is represented in red color. So in the sorting image, you can see we are sorting 3 different quadrants, which is CD34 negative, CD117 positive, the left upper quadrant, the right quadrant, right upper quadrant represented by CD34 and CD117 double positive, the lower right quadrant represented by CD34 positive, CD117 negative. All 3 quadrants are sorted, and the double negative, which is the lower left quadrant, is not sorted. So after sample sorting, the sorted cells are rerun again through the cytometer to obtain a profile to gather information on the purity and percentage of sorted cells. The post-sort profile of CD34 versus CD117 shows the purity of the sorted cells. The 3 quadrants, the double positive and the single positive, shows 99% purity of sorted cells, which is represented by the red color. The lower left quadrant, CD34 negative and CD117 negative, shows that the cells that are not of interest is not sorted. So by this way, we can obtain high purity of the cells of interest, which can be used for further analysis. So flow cytometry has still many challenges that needs to be addressed. It is a very versatile platform for characterization of different cell types in the support of clinical and preclinical studies. In a regulated environment, a quality control or QC sample that mimics the actual study samples are included during analysis to monitor the performance of a method and to assess the integrity and validity of the results. There are very few commercially available preserved whole blood samples that can be used as QC for certain markers, but has a short shelf life. One of the options to consider is, can a sample be fortified with a defined amount of antibody that can give similar amount of fluorescence intensity, counts, and percentages of parent population at every use? This option needs to be further investigated. More challenges that we see is when handling diseased state population. Obtaining fresh diseased state population is challenging depending on the logistics. Assessing stability for diseased population samples are very challenging, and it brings into question if a biomarker signal enhanced by using stimulants to emulate disease state can be used as a surrogate QC, which can be used to compare stability results. More standardized stability assessments can also pave the way for multi-site clinical studies to acquire results with minimum variability following the same sample handling and processing procedures. The standardization of stability assessments for flow cytometry needs to be also further investigated to ensure similar results across sites. I thank you all for joining the webinar. With that, I will turn it over to Lorraine for questions.
Thank you so much. That was really, really educational. We do have a few questions that have come in during the time, so we'll go ahead and address those. Number one was, why do we need different types of techniques for PBMC isolation, and are there any issues with red blood cell contamination during PBMC isolation?
So the different kind of techniques that we discussed about, the overlay, underlay, and separation techniques, these are three techniques that are commonly used. The overlay and underlay techniques are manual methods and is not widely used as much anymore. The separation tube technique is the one that is more commonly used, as it can provide distinct layers of PBMC without any kind of red blood cell contamination. So with using separation tube techniques, the red blood cell contamination is very minimized. If we do see red blood cell contamination, some procedures, depending on the downstream application, may require red blood cell lysis to further remove red blood cells. Using Muse cell analyzer for counting eliminates the red blood cells from the counting and viability information, as the red blood cells are non-nucleated cells and they are not part of the population of interest and they occur below the threshold line. Therefore, we have not seen much red blood cell contamination, especially using the separation tube technique, and we are able to obtain cleaner PBMC counts and percent viability. Furthermore, red blood cells die during the freezing process, which can also help with downstream applications.
All right, thank you very much, Anamica. The next question refers to Celerion capabilities and asks the question, what is the throughput for flow cytometry sample analysis at Celerion?
So the throughput depends on the amount of samples that we can acquire during a particular period of time for a particular assay. The throughput is typically determined during our method development and validation process based on the post-process stability that we can establish. If we can establish a stability of 24 hours, we will be able to do batch sizes of 26 to 28 samples. The examples that were shown previously for the CD34 and the TBNK assays, we have established that we can do a batch analysis of 28 samples along with controls, which can be acquired within 6 hours of staining. If the staining is performed in a 96 well plate, we can acquire 48 to 96 samples, depending on the number of replicates analyzed for each sample.
All right. And then the final question we have is, how and what data will be received by the clients from flow analysis?
After flow analysis, we do get a PDF file from our software that lists the gating strategy that was performed for each and every sample. We also have an Excel data analysis to calculate the absolute cell counts, percent inhibition, etc., depending on the assay. So we can provide both the raw data, which is the gating strategy, as well as the Excel data with the raw numbers for the clients. It can depend on client needs. We have some clients that require both, and we have some clients that just require the final numbers from the population. And these can all be defined when the contracts are signed by defining the criteria in a data transfer agreement.
All right. Well, that's very helpful, and thank you. This actually brings us to the end of our webinar, and as a reminder, a copy of the webinar will be available on our website, and we will address any remaining questions directly within 24 to 48 hours. And thank you very much for attending the webinar.